Hi, everybody. My name is Olivia, and today I will be discussing a study that examines growth hormones' effect on lipid accumulation in skeletal muscle during prolonged fasting. So, what exactly is this study testing? So, lipid accumulation in the skeletal muscle is a physiological adaptation that occurs in a state of starvation or, in this case, a state of prolonged fasting. Um, and growth hormone secretion amplifies while in a state of fasting and promotes lipolysis and lipid oxidation. Lipolysis is the metabolic process through which triacylglycerols, which are tags, break down into glycerol and free fatty acids. So lipolysis is just the breakdown of fats, basically. Um, and It's unknown to which degree that lipid deposition and metabolism in skeletal muscle during fasting depends on growth hormone action. So, some acronyms that should know because they are discussed throughout the study would be intramyocellular lipid, which is IMCL. IMCL is just fats that are stored in the muscle cells. During fasting, which are used as metabolic fuel during exercise as well. Accumulation of IMCL appears to be a physiological adaptation to fasting, regardless of one's body composition. So, growth hormone, I'm sure we're all familiar with it. Growth hormone helps to maintain normal body structure and metabolism, including regulating insulin and blood sugar and keeping it within a healthy range. Growth hormone triggers fat cells to empty their stored energy, and this is the opposite of what insulin does. Insulin causes us to store fat in our cells. And then growth hormone antagonist (GHA) is basically it just blocks the secretion of growth hormone from the pituitary gland. And tags or triacylglycerols, I already mentioned, they're just the main component of human fat. They're mostly stored in adipose tissue, and so basically, they're just the main storage form of fats within the body. So the hypothesis of this study is basically evidence suggests that growth hormone directly regulates skeletal muscle lipid metabolism and IMCL content. Um, Growth hormone is an important regulator of insulin and metabolism in skeletal muscle. Um, so there's studies where treatment of growth hormone reduces overall fat mass and decreases tag synthesis. Um, and growth hormone production is increased during fasting. Um, so is it the increase of growth hormone production that occurs during fasting that regulates lipolysis and IMCL deposition that occurs during fasting, or would these processes still occur independently of growth hormone? That's basically what the study is testing. The subjects included in this study are 9 healthy, overweight and obese men with a median age of 24 years and a mean BMI of 30. Um, they're overweight and obese, but otherwise they're healthy. They don't have like they don't have like diabetes or anything like that. Um, Obese subjects were chosen because they have higher fatty acid fluxes during fasting than lean subjects. The method that was used was a randomized placebo-controlled crossover design. Um, it used the same 9 subjects, and there was 3 different studies on these subjects with 4 weeks in between each study. And the control group was um, observed during an overnight 12-hour fast. The second group, called fast, they were observed during 72-hour fasting, and they were given a placebo to mimic the growth hormone antagonist. And then the third group that was studied, well, it's the same group of men, but the third time that they were studied was during a 72-hour of fasting in combination with GHA. And with this method that they use, it will provide an answer to whether or not 72 hours of fasting does IMCL accumulation in the skeletal muscle occur in the group who is not synthesizing growth hormone due to receiving a GHA. So the hypothesis is basically that growth hormone is possibly what causes the skeletal muscle to shift from burning glucose to lipid oxidation for energy. So the main question here is, will this metabolic shift still occur without the synthesis of growth hormone? So I just want to talk a little bit about what happens to our bodies during fasting. So in a state of starvation or prolonged fasting, when blood glucose is depleted, the body will convert to using fat stores as its main source of energy while conserving protein. Um, Lipolysis of tags produces glycerol and free fatty acids. The glycerol that's produced from lipolysis is a source of carbon for gluconeogenesis. Gluconeogenesis is when the body creates glucose from sources other than carbohydrates. So even when we are not consuming carbohydrates, whether it's just due to a low carb diet or um, in a state of fasting, the body will create its own form of glucose, which I found that very interesting. Um, so just continuing on what happens during fasting. Free fatty acids are transported and either oxidized in tissues by a process called beta oxidation, or they get converted to ketone bodies to be used as fuel for the brain, which decreases the consumption of the already depleted blood glucose. And in a fasted state, white adipose tissue releases free fatty acids to use as fuel elsewhere in the body. So the measures taken in this study, the researchers measured IMCL deposition using magnetic resonance spectroscopy, MRS, and blood samples were taken for plasma metabolomics. Sorry, I can't say that very well. Assessment. Um, and then muscle biopsies were obtained to measure regulators of substrate metabolism, and body composition was also observed. The findings were that lean body mass and total body water was reduced in both of the fasted groups, not affected by the growth hormone antagonists. The increase in circulating free fatty acids, growth hormone, and ketone bodies was found in both of the fasting groups, independent of the growth hormone antagonist. And there was a decrease of insulin levels during fasting in both of the groups. And if you look here, pretty much both of the groups, with the fasting and the placebo and the fasting with the antagonist here, um, compared to the 0 hours of fasting, they all increased. Um, they all increased except for insulin decreased. So the antagonist didn't really have any sort of effect on any of the variables that were being measured during this study. Another thing was that branched chain amino acids and sulfur amino acid metabolites increased during fasting. So BCAAs stimulate the building of protein in muscle, and they possibly reduce muscle breakdown. There are some studies that suggest that BCAAs have a protein-sparing effect. So this suggests that in a state of fasting, your body is actually preserving your muscle. And then the sulfur amino acid, um, these amino acids are they contribute substantially to the maintenance and integrity of cellular systems by influencing cellular redox state and cellular capacity for the cell to detoxify toxic compounds like free radicals and reactive oxygen species. So sulfur amino acids are helping the body to detox at a cellular level. Furthermore, the findings, so fasting is associated with elevated levels of intermediates from both beta and omega oxidation. Like I said, beta oxidation is the process by which fatty acids are broken down to produce energy, and omega oxidation is an alternative oxidation pathway when beta oxidation is blocked. Um, so basically, since both of these processes were used during fasting for these obese men, it suggests that beta oxidation may have been saturated because there was a massive supply of lipid intermediates um, that were being oxidated. So the body used omega oxidation as a backup because beta oxidation was saturated. So the results, like I said, growth hormone was elevated in both groups, despite the one group taking a GHA. There is also an increase in circulating free fatty acids, glycerol, ketone bodies, which indicated an increased supply of lipid intermediates to skeletal muscle. The study also found that fasting resulted in an increase in IMCL content by about 75%, suggesting that lipid uptake in skeletal muscle increases during fasting and that this favors IMCL deposition. And there wasn't really a detectable effect of GHA on skeletal muscle lipid metabolism. So, in conclusion, fasting induces lipids as a source of fuel in the skeletal muscle. So, lipids accumulate in skeletal muscle during fasting, which is not modified significantly by GHA, which could indicate two things really. It could indicate either there is insufficient growth hormone blockade, or it could also indicate that the body has multiple mechanisms to ensure lipid mobilization, meaning that growth hormone isn't the only regulator of lipid accumulation in skeletal muscle during fasting. This also suggests that lipid accumulation in skeletal muscle during fasting could be driven by an increased supply that exceeds the demand for lipid utilization in obese subjects, because obese subjects have more tags. So the result could be different if the subjects were lean subjects. And then the limitations of the study were that the sample size only included 9 subjects. It only used obese subjects, and I think that lean subjects could have a very different effect, or fasting could have a very different effect on lean subjects, and only male subjects were used, and females have a very different hormone cycle than males, so I think fasting could potentially have a very large difference on um, females. And there's potential that not enough GHA was given to the subjects because um, growth hormone was produced regardless in both subjects, so in both groups. So, and then here is my bibliography. All right, well, thanks for watching.